Hi, my name is Jim Heath. I'm the president of the Institute for Systems Biology and a professor here. And I'm here today with Professor Wei Wei, my colleague. And we're here to talk about a paper that we both just had published in Cell Systems. It's entitled Signaling and Transcriptional Dynamics Underlying Early Adaptation to Oncogenic BRAF Inhibition. So, as with most scientific papers, it's a bit of a mouthful. And I think before we start, I want to make sure I give acknowledgement to Chunmei, Cam Flower, and Forrest White. Forrest White and Cam were from MIT and were important collaborators in this work. In fact, we couldn't have done it without them. Okay, so that having been said, so BRAF inhibition is that's a common drug that's used in melanoma.
Right.
And it's been known, it was like a miracle drug when it first came out, and then over time it was found that while it does actually eradicate melanoma tumors in BRAF mutant patients that have BRAF mutations,
Right.
most patients develop resistance to it, but they do it over a period of maybe few months or a year.
Yes, or a year, right.
And in this paper, we studied the dynamics of BRAF inhibition over the period of like hours and maybe a couple of days.
Yeah.
So what do you get when you study such short time dynamics? What's the point of that?
Yeah, sure. So I think basically the resistance to the BRAF inhibitor, a common targeted therapy for metastatic melanoma, is actually what we call the drug adaptation process. And drug adaptation in cancer basically refers to the ability of cancer cells to survive despite being exposed to treatment that are meant to kill them. So unlike the genetic mutation that cause permanent resistance over time, drug adaptation happens much faster and is temporary, flexible response that allows some cancer cell to tolerate the drug effects before permanent resistance sets in. So you can think of it like a survival instinct. When cancer cells encounter a targeted therapy, the drugs initially block the cancer driving proteins. In our case, it's the BRAF. However, instead of dying immediately, the cancer cells activate alternative survival pathway like flipping backup power switches to keep essential process running. And these changes don't require new mutations. They only involve like rewiring the ways the cell's existing protein or gene function. So this study basically, we challenge a common belief about how cancer cells develop resistant targeted therapy. Many people think, the drug adaptation emerged only after like a prolonged treatment after a week or even several weeks. But our research revealed that melanoma cells begin adapting within hours of drug exposure. And here we use like mass spectrometry-based phosphoproteomics and deep RNA sequencing to capture thousands of signaling events in the first minutes, hours, and days after like drug exposure, as Jim just mentioned. And surprisingly, we found that the melanoma cell don't passively shut down the drug effect. Instead, they quickly rewire the internal signaling network to survive within just a few hours. And they activated a group of protein we call a Circ family kinases, which help them survive therapeutic stress. And importantly, we also found that blocking this early adaptation with another drug called deceptinib significantly improved the treatment effectiveness in both like lab-grown cells and animal models. So this is important because if we can really understand how cancer cells adapt at very early stage, we can develop smarter combination strategies that can cut off the cancer cell survival strategies before they take hold and potentially leading to a more durable treatment for melanoma patients and also for other cancers.
Okay, so let me maybe recap a little bit of that.
Right.
So oftentimes, and at least traditionally, when cancers would develop resistance against a drug, I think many scientists viewed it as sort of a Darwinian selection,
Right.
that you would have cells that would gain new mutations. They would divide, and new mutations would actually give the cells the ability to circumvent the effects of the drug.
Yeah.
But of course, when you have Darwinian selection, what that means is that you had a very small subpopulation of cells that wasn't responding to the drug,
Hmm.
that had the mutations, and over time, those cells just grow out and dominate because the other cells are affected. So that's
Yes.
Darwinian selection. That's not what this is. This is this
Right.
saying that the same cells that respond to the drug are the ones that get resistant to the drug. That's adaptation.
Exactly.
Happens very
Yes.
fast.
Haven't verified.
And
Yeah.
exactly. So now something that's, I think, kind of striking in this paper
Yeah.
is that when you look at BRAF inhibition, as a sort of a cancer biologist, you would expect, there's a well-defined what's called signaling pathway of maybe
Yeah.
half a dozen proteins that are associated with inhibiting BRAF. And the way that one would oftentimes see the activity or inactivity of those proteins is by looking at the phosphorylation status, which of course
Right.
that's known. In this paper, we looked at thousands of phosphoprotein sites.
Yeah.
Um, so did they all change? I mean, maybe you could comment on how does that global view change the thinking about what happens in these cells?
Right, right. So I guess the reason we were using kind of a omic level approach, the phosphoproteomics, instead of just looking at specific proteins, is that we really want to reveal a full picture of how the cancer cell adapt to the BRAF inhibition and from the comprehensive signaling perspective. So that's the reason we were analyzing like thousands of proteins, and we do see already uh, what people have already known about. For example, when the drug hit its target, you see all the downstream signaling in proteins gonna shut down by the drug effect. However, the melanoma cell don't just sit back and just take the hit when we block the BRAF with the BRAF inhibitor. Instead, they quickly start looking for alternative survival routes. And these survival routes, there are many different survival routes, but just within a few hours, we found that the most significantly elevated one is the group of protein called subfamily kinases, which essentially like serve as a kind of a backup survival mechanism when the primary BRAF-driven growth pathway is shut down. And what's even more fascinating is about how this adaptation happens. So by looking at like thousands of phosphoprotein sites and also transcriptional regulations from the RNA-seq as well, it turned out that these and treating these cells with a BRAF inhibitor actually causes a buildup of the reactive oxygen species, a form of a molecular stress. This oxidative stress triggered this subfamily kinase signaling, giving cancer cell a way to survive and despite the drug presence. So in this way, the very process of blocking the BRAF unintentionally actually set off a chain reaction that helps the cell escape. And the phosphoproteomics actually help us to reveal and to resolve this chain reaction step by step, using this time series data from minutes to hours to days.
So, a striking conclusion of this is that even though it may take a patient several months or a year to develop resistance against a BRAF inhibitor,
Right.
the seeds of that resistance happen within the
Yeah.
first 24 hours, really.
Exactly. Yeah.
And you can actually predict with high accuracy that it's going to happen. Uh,
Exactly. Yeah.
so another striking aspect of this paper was that the whole process, you take the drug away, the whole process is reversible.
Right.
And you just
Right.
go right back to the cells that as if they were drug naive.
Mm-hmm. Yeah.
Um, and I think that type of reversibility, certainly you would never see that in a Darwinian selection process. But in these
Right.
adaptation processes, nature has the ability to turn the clock backwards.
Yeah. You're right. Yeah.
Um, okay, and so, finally, this paper is, I think of it as one of two. And so there's another one coming out, hopefully pretty soon, and
Yeah.
where, maybe just give a little teaser of what's looked at in that next paper, and we can revisit and talk about it when it comes out.
Right. So yeah, I think the one of the most interesting aspects of our finding in this paper is that when we remove the drug, the melanoma cell didn't simply reverse their adaptation by like retracting their original steps. And instead, they follow a different path back to their original state, much like what we see in hysteresis in physics. So the other paper is actually we also interrogate the same symptom for a much longer period, over a month. And we also use time series transcriptome and epigenome studies to really track step by step how they evolve over time and eventually become purely like drug resistance. But when we release the drug, the cell can also revert back to the original state, but follow a different follow a different path. So how to think about that? Actually, as an analogy, we can think about that as the hysteresis in magnetization. So, for example, when we apply a strong magnetic field to a piece of iron, the item inside the iron, align in certain ways, and the material become magnetized. But when we remove the external field, the iron doesn't immediately return to the original state, right? Instead, they may retain some magnetization due to a kind of memory of the applied force, and it's take a different path back to the equilibrium if you try to demagnetize it. So in our study, we actually see a pretty similar kind of biological hysteresis in how melanoma cell transition back after drug removal. And initially, when we expose them with a BRAF inhibitor, they went through distinct phase transitions, gradual shift in their molecular states as they adapt to the stress and of the drug. However, when we took the drug away, instead of simply reverting those steps in order, the cell take an entire different molecular route back to their pre-treatment state. This suggests that the cell doesn't forget the experience of the drug exposure. They retain a kind of molecular memory that shapes how they recover. And this hysteresis phenomenon not only happened in short-term treatment, as we present in this study, but also happened in a longer-term treatment as well, as demonstrated in another study Jim just mentioned, hopefully coming out in the next few months. And this finding is important because it tells us that adaptation is not simply an on-off switch. It's actually a more complex dynamic process where the history of drug exposure influence future cell behaviors. And if we can decode this memory effect, we might be able to exploit therapeutically. For example, if whether we can design treatment that force the cell into a vulnerable attractor state from which they can't easily recover, and then we may hit the cells with the hit vulnerability with another drug to achieve a more synergistic drug effect.
All right. Well, thank you, and congratulations again to both of us.
Yeah, thank you, Jim.